And so in the second part of this lecture, we're going to focus on this M, or mitotic phase of the cell cycle. And so you've likely heard and learned about mitosis before, and so this is mostly going to be a review for many of you. What I hope to add in is a little bit more about the regulation between the phases for mitosis. And so what mitosis really is the division of the DNA, or the chromosomes, from one mother cell to produce two identical daughters. And so in mitosis, not only is are the chromosomes split, the cytoplasm or the cells are split as well. And mitosis is divided into 5 stages for the purposes of this class: prophase, metaphase, anaphase, telophase, and then cytokinesis, which is the physical division of the cytoplasm. And I'm going to talk more about each of these stages in more detail in the next couple slides. And so the first stage of mitosis, the replicated DNA as chromatin needs to condense in order to form chromosomes. And this is important because ultimately you're going to organize this DNA and then try to split it evenly between 2 new daughter cells. You can imagine that trying to do that with 6 feet of chromatin that's unwound and kind of stringy and loose within the nucleus would not be terribly easy. And so by organizing the chromatin into chromosomes, that division of DNA that happens later in mitosis can happen much more easily. And so you can see here an interphase cell, or a cell that was just leaving G2, and it has kind of a loose chromatin in the nucleus, which condenses into chromosomes you can actually visualize in a prophase of the cell. And in addition to the condensation of chromosomes, the nuclear envelope that normally surrounds DNA disappears so that the chromosomes are free to move about the cell. Some of the organelles fragment to also move out of the way of the DNA of the cell, and the mitotic spindle begins to form. And the mitotic spindle is an important structure that will ultimately divide these chromosomes later in mitosis. And what the mitotic spindle is a structure made out of the cytoskeletal elements, particularly microtubules, and it's responsible for dividing chromosomes during mitosis. And so you can see here a cell whose chromosomes are labeled in blue and whose mitotic spindle is green. And the mitotic spindle organizes during prophase and emanates from a particular location called the microtubule organizing centers, or MTOCs. And you can see that dark green fluorescence, or that bright green dot, on 2 sides of the cell, or at the 2 poles of the cell, and many, many microtubules emanating out from there. Microtubule organizing centers have different names in plant and animal cells. In animals, they're known as centrosomes. In plants, they are centrioles. And so that mitotic spindle has to organize and then grow during prophase. And once the chromosomes have been condensed, the mitotic spindle has been formed, the chromosomes can actually attach to the spindle fibers, or to those microtubules. And that happens during the next phase of mitosis, which is known as metaphase. And the chromosomes are attached to the mitotic spindle at a particular place on the chromosome, directly in the center of sort of that canonical X shape you think about chromosomes having, at a structure called the kinetochore, which is inside the centromere, or the portion of chromosomes that are in the center. And so once the pieces, or the mitotic spindle, has attached to the kinetochores of chromosomes, that mitotic spindle kind of pushes and pulls those chromosomes for a long time, up until they are perfectly lined up in the center of the cell, or the middle of the cell, which is known as the metaphase plate. And so you can see here the cell in prophase with its condensed chromosomes, and mitotic spindles actually in red. And then in metaphase, that mitotic spindle has pushed and pulled those blue chromosomes and lined them up directly in the center of the cell. And the same can be seen up here in this image on the top right. The mitotic spindle is in green. Those chromosomes, again, are in blue, and there are 2 poles, or two microtubule organizing centers of the mitotic spindle, and those fibers have pushed and pulled the chromosomes till they're lined up perfectly in the center of the cell. And so when you look at a cell, this line of chromosomes down the middle is a hallmark that that cell is in metaphase. And so the transition from metaphase to the next phase of mitosis, or anaphase, is one of the most highly regulated parts of the cell cycle. And I'd like for you to take a second and pause the video and think about why that might be. Okay. And so in metaphase, the chromosomes have lined up in the center of the cell. And then in anaphase, that is where the chromosomes start to move apart towards what will ultimately become two new cells. And you can imagine that if the chromosomes are not perfectly aligned in the center, there might be some that are still kind of on the left hemisphere of the dividing cell, or some that are on the right. And when the mitotic spindle starts to pull those chromosomes apart, instead of it being an equal division of genetic material, because it's divided right down the center, there can be an improper number of chromosomes pulled or moved into each new dividing cell, which can lead to too much DNA in one cell, too little in another, and too much or too little DNA can ultimately cause issues later on. And so this metaphase to anaphase transition is highly, highly regulated. And it's regulated by a particular complex known as the anaphase promoting complex, or the APCC. And so anaphase promoting complex, or APCC, is a particular type of enzyme known as ubiquitin ligase. And a ligase is something that adds a molecule to another molecule, ligates things together. And since APCC is a ubiquitin ligase, what it does is it adds a ubiquitin molecule onto this protein here called securin. And securin's job is basically to hold an enzyme called separase in check. Separase's job is to cleave cohesin molecules that hold the sister chromatids of a chromosome together. And so cohesins act sort of like little clamps, and they hold the two arms of a chromosome, or the two sister chromatids, together. And separase is really effective at opening those clamps, cleaving those cohesin molecules, and separating sister chromatids. And since we said we don't want to do that until the exact right moment, when all the chromosomes are lined up during metaphase, securin exists to keep separase inactive and in check and not degrading cohesin, so that the sister chromatids are held together until the correct right moment, right? And so what APCC does to promote anaphase is to ubiquitinate, or add a ubiquitin to securin. And whenever you add a ubiquitin molecule to something, it basically targets it for degradation at the proteasome, or it basically targets it to be destroyed and broken down. And so when APCC ubiquitinates or adds a ubiquitin to securin, it targets it for degradation, and that leaves separase free. And now separase, as an enzyme, can go and do its job. It's active, and its job is cleaving those cohesins. And when it does that, it basically cuts right down the center of the two sister chromatids and allows them to separate in the next stage of mitosis, which is anaphase. And so after APCC ultimately leads to separase cleavage of cohesins between sister chromatids, those sister chromatids that were lined up nicely at the metaphase plate can be pulled towards the poles of the two new cells, towards either pole of the cells. And the way that they do that is by the microtubules of the mitotic spindle shrinking towards the centrosomes, or the microtubule organizing centers, and basically dragging those sister chromatids along with them. And so you can see once again here, all of those spindle fibers in red, and the chromosomes moving towards the poles in blue. And then all good things must come to an end, right? So after anaphase has separated the sister chromatids, the chromatin can begin to decondense. And the two new nuclei of what will ultimately be the two new daughter cells can reform. Organelles can begin to reform, the mitotic spindle is no longer necessary because the DNA has been divided, and so it can dissolve, right? And so if you'll notice some of these hallmarks of telophase, which is that phase following anaphase, or the separation of sister chromatids, is that they're all basically the exact opposite of what happens in prophase. So in prophase, you get ready to divide, and in telophase, you basically get ready to become two new cells. The chromosomes decondense back into chromatin, nuclei reform, organelles reform, and the mitotic spindle is no longer needed, so it dissolves. But what you'll notice here in the center of these two cells, or this cell in telophase, is that these two, what will be new cells, are still physically connected. And so there has to be a way to separate that and those two new cells and create the daughter cells. And this is the final phase of mitosis, or cytokinesis. Cytokinesis is the physical separation of daughter cells. The DNA is already been separated at the end of telophase. And so now we need to split the cells apart. And since the focus of this class is mainly animals, we're going to talk about animal cells, which have a pretty squishy cell membrane that can actually pinch off. This cleavage furrow is formed between two new cells. It's made up of, there's a contractile ring made up of actin and myosin that basically contracts and pinches tighter and tighter between the two cells until ultimately dividing them from each other. And at the end of cytokinesis, you've effectively made two new daughter cells from one mother cell.